Today, we'll walk through an example of how you all can use BioFinder in your own research and your drug discovery efforts. So we wanted to really follow the model of SciFinder in introducing BioFinder. SciFinder, our chemistry product, which is used across the country, across the world, across the entire chemistry industry, really as a single reliable source of reference information for as many different chemistry questions as possible. And so we really, with BioFinder, wanted to bring that philosophy to the life science realm, but adapt the experience as appropriate for biology. So as you all know, the key research in life sciences comes not just from specific properties of small molecules, specific proteins, but more so really the connections between those entities, the broader context around how those molecules interact with one another, how they assemble into pathways, how those pathways play roles in larger biological processes, how those processes play into disease progression and development. So we have to bring together data from many sources, many different domains, and we have to make all those data work together to give you the full picture. So we assemble all of this information into a massive knowledge graph, and through BioFinder, we help you to parse through different slices, different angles of the data as a whole, while trying to orient the context around your specific area of interest. So for example, if you're interested in small molecule ligands, BioFinder can present information like their structure, their chemical properties. We show information about proteins, their structure, their properties, sequence information, but we also show the broader context. We provide information around the interaction between them. We present bioactivity data. We really want to show the underlying data behind ligand-protein interactions, what roles those proteins play in what processes, what roles do they play in disease. So each of these major entities, ligand, protein, disease, they can all or each be an entry point into the broader knowledge base. And of course, we always want to incorporate document data too, so we can always trace that information back to their original publication sources. So by bringing all these data together, we can bridge the gaps between disease, target, treatment modality. We have comprehensive information on small molecule ligands, antibodies, antibody drug conjugates, information on their physical properties, as well as pharmacology information on their interactions with their target proteins. We've also assembled one of the largest collections of disease biomarkers in the industry, and we've created a unified disease ontology to link those protein and disease data together. We've also extended our expertise in small molecule chemistry to protein chemistry, and we've created a protein ontology that we can then use to connect information on proteins, genes, pathways, sequences, really create a comprehensive view of your target of interest and the surrounding biological context that you'd be interested in. So yeah, really just taking a holistic approach to life sciences information, making all of that data searchable, accessible to you as you go about your research and your workflows from day to day. And then on top of this data foundation, we're building multiple flexible methods of navigating that information within our content corpus. So the content is always going to be the foundation. Then on top of that, we've built the BioFinder interface, which gives you a number of self-directed ways to explore, slice and dice the information in our knowledge graph as you see fit according to your interest. And then on top of that, we've built a ScienceSmart AI layer that can surface, summarize information through a very friendly and flexible natural language interface. You can even have conversations with it, if that makes sense. And so we'll show that off a little bit more in a few minutes. But regarding Newton itself, our AI assistant that we're calling Newton there are a few things that we think set it apart from other players in the industry. So first and foremost, it's that, again, that CAS knowledge base that we use as our foundation. So Newton's outputs will always be grounded in this curated data set of chemistry and biology. Then on top of that, we didn't just take an off-the-shelf language model, slap it on our database, and call it a day. We really did the work to stitch together a number of different agents, subagents, different specific tools that are tuned to reason through your query and produce as high quality of response as possible. So then, because our content are always grounded in our document knowledge base, those responses that are generated by Newton are always going to be traceable back to the original source material. So that's going to be really valuable in verifying the output and making sure we avoid hallucinations that can put you off track. So that's really the value, the benefit of the BioFinder platform. We're leaning on our expertise of bringing in information from disparate sources, curating them, aligning them with each other, displaying that information with a proper context to you, the users. So you don't have to spend all the time hunting down different information in different individual sources. We try and bring that to you all in one place, make it intuitive so you can find the answers that you're looking for, and then get back to the bench. So I'd like to show you how we can do that right now. Okay, is that showing for you all, Christy?
Looks great.
Looks great. Okay. Awesome. So yeah, just switching over to BioFinder here. So this is the BioFinder homepage, for those of you who are not familiar. So again, we try and present you with the tools to explore in whatever way that makes sense for your workflow and your interest. So you can select, you can do keyword search, you can select from a number of search modalities. So there's ligands, there's scaffolds, proteins, diseases, if you're looking for something specific, or you just want to query the knowledge graph directly. Or, as I mentioned, we have our CAS Newton Research Assistant accessible through this tile right on the homepage. And so, yeah, we just want to give you the options right off the bat as you log in to make the tool as flexible as we can for whatever you need. So for the purposes of this demo, I'm looking to get smarter in an area that maybe I don't have a lot of expertise in. So I want to get a summary of a lot of information relatively quickly. And so for that use case, Newton is really great. So I'm going to start with clicking into CAS Newton here, just right on the home page. And it'll present you with a search tile, search bar right here, where I can ask whatever question I'm interested in. I can choose to draw a structure here if that's how I want to engage with the assistant. But for the purposes of this demo, I'm going to actually just ask it a question. And since Newton, again, our CAS knowledge base is quite substantial, Newton is going to be combing through a number of different data sets. So I have a preloaded example in my search history on the sidebar here. So let's take a look at that. So let's say I'm just interested in non-small cell lung cancer as a disease area. Let's say I'm coming into the project as a relative novice. I'm looking to start very broadly, get up to speed about a disease area that I might not be very familiar with. Let's say it's my first day on a new project. I'm just looking to dive in. So I'll start by entering a prompt, asking Newton a question to get an overview of the disease. So I'll ask, give me a overview of NSCLC, tell me about the disease. Of course, Newton's going to understand that NSCLC is standing for non-small cell lung cancer. We'll ask it about the underlying mechanistic causes, the key proteins and pathways involved, and then ask Newton to summarize the various strategies for treating the disease and provide details on how effective they are. So this will give me a really nice overview of the disease area. since I'm interested in developing some potential novel compounds to help treat the disease. I'll want to start broadly, broad knowledge of the disease in general, then narrow down to the specific existing treatments, how they work, and then the mechanism that they're leveraging in between. So again, just to help me inform my own decision making when designing a new compound or treatment strategy of my own. So this is the summary it's going to generate. It'll start by defining the disease, non-small cell lung cancer, epithelial lung cancer. It'll break it down into the major subtypes here, and then give information on staging. It'll get deeper into the underlying mechanisms. We'll see that Newton has organized the mechanisms into different oncogenic drivers, tumor suppressor genes, immune evasion, environmental and epigenetic factors as well. So I can see the major drivers here are KRAS, EGFR on the oncogenic side, tumor suppressor genes, loss of tumor suppressor genes like p53. We'll see downstream activation of RAS, PI3 kinase, growth, proliferation related pathways. So that all makes sense. And then we'll get into treatment strategies. So now at this point, I know the proteins that are involved. I have a sense of the potential targets and the role they play in their respective pathways. Now I can start looking at how those may be targeted from a therapeutic perspective. So we can see, again, the response is broken down, organized into treatments, into EGFR inhibitors of different targets, EGFR, ALK1, ROS1, KRAS inhibitors, MET inhibitors. So let's just actually stop here, and we'll just pick one of these therapies to go do a little bit of a deeper dive. So it lists Osimertinib right here, a third generation EGFR inhibitor. That sounds pretty interesting to me. So let's click in. And so now this will take me to a detail page that brings together all the information that we have for Osimertinib or whatever ligand that you might be interested in. So everything that we have, again, pulling together, giving it to you in one place. You don't have to go hunting around in different areas of the application or even going out and using different tools online that you might have access to and hunting and gathering for the information you need. We try and bring everything together in one full picture right off the bat. So on the first page here, we'll give you a summary tab of Osimertinib. Of course, the chemical structure. We'll give you a drug intelligence summary that shows the status, the indications, primary targets. I can scroll down and see all the various chemical properties, synonyms, SMILES, all that standard information that you would expect. But of course, we're interested in binding information, how effectively this treatment can bind to its protein target. So we have pharmacology data organized right here on this tab. I can click over, scroll down, see here's all of the actual bioactivity assays that we've collected from numerous publications. So as expected, most of these are going to be measuring binding with EGFR. I can click view under the assay column to view assay details. It'll summarize the assay as it's described within the publication. So it'll give me the value, the procedure, parameter, so forth. So I can view all that right here, pulled out of the document. If I'm interested, I can go back to the source document. So click the reference link here, and this will give me a summary of the paper that we've pulled out. I can get more detail about the specific publication, as well as a readout of all the measurements that we've pulled out of that specific document. So I can click back over to my ligand information here, and I'll just show Again, the breadth and depth of additional information we've taken out regarding your particular ligand of interest. So um, we have information on ADME and toxicology. I can view all of the proteins that are associated that are known to bind with osimertinib. I can click into the diseases tab here, show you, it'll bring together all of the diseases that are associated with osimertinib, that are associated with treatments for these diseases. We have clinical trial adverse event information cataloged here as well. So I can sort by status, by phase, by sponsor. So I can click through and you'll see here's all the trials organized by sponsor. So most of them being run by AstraZeneca. So of course that makes sense. Um, I can also view information here about metabolites for this given ligand. This will bring, we have information on both known and predicted metabolites. And I can click in here for a known metabolite. It'll give me more information about the actual enzymes that are involved in catalysis, the organism, and then the source from where this information is coming from. So really, again, trying to bring a comprehensive view, giving you everything that you need to act on your experiments. Um, I can also dive into more information about the protein targets. So in this case, EGFR is the major one here. Um, so I can click EGFR, and now we've kind of taken a lot of the same information, but now reoriented it around the protein. So this is now a protein-centric view for my protein of interest, EGFR. Again, it'll bring me to a summary page. It'll give the 3D structure that we've pulled from the protein database. It'll give a summary of the protein and gene function here. Again, synonyms, external links to databases you might be interested in. We have a number of different crystal structures that you can scroll through. Pharmacology data, much like the ligand detail page, we organize the pharmacology information that we have cataloged. But now, where if I was looking at the osimertinib page, it was showing me a list of all the proteins that are binding to osimertinib. Now, since we're looking at protein information about EGFR, it's showing all the ligands that are known to bind with EGFR. So again, we're reorienting the data given the proper context here. Here's all the ligands that we've cataloged as interacting with EGFR, the important diseases that EGFR might be involved in, and we even have information on pathways. So I can click into a specific pathway here. It'll give me a step-by-step breakdown of the pathway steps, and we even have sequence information as well. So whatever has been identified as kind of the canonical protein sequence, the canonical transcript, we're going to present the sequence information to you right off the bat, and then we make it as flexible as possible. You can run a blast search, you can download that sequence in a FASTA file, you can copy it to your computer's clipboard. And then if you're interested in alternative transcripts, alternative isoforms for the protein, we provide those to you as well. I can just click into here. Here's the sequence information. Again, I can take that sequence and do additional steps downstream for whatever I'm interested in. All right, so yeah, again, the theme here, just trying to bring together an abundance of information from different sources, organizing that data around the correct context, showing you as the users the information we have for a given ligand, protein, disease, wherever your interest may lie. We're trying to provide you the proper information. So let's click back over to Newton, continue going through this response here. So Newton is describing the various EGFR inhibitors. Says here, osimertinib is a third generation EGFR inhibitor. So as I'm getting up to speed about this disease area, the various treatments involved, that makes me wonder, so what about the drug design is going to make it make it better than previous treatments? So that's actually another really good question for Newton. And so I've gone ahead and asked Newton. I see Osimertinib is a leading EGFR inhibitor. How does it compare to previous treatments, and what makes it better? So um, just again, I've got the response preloaded here, but in an actual use case, I would just be clicking down into the search bar, asking a follow-up question, and then it'll generate another comprehensive response. So now it'll start by getting into the first generation of inhibitors, gefitinib or erlotinib, give a summary of each of these. It'll explain that this is a class of tyrosine kinase inhibitors. They work by preventing phosphorylation, halting downstream pathway activation, preventing cell growth and proliferation. All great information to have as I think about my own potential treatment strategy. Okay, but I can see here's the key limitation. So there's acquired resistance after a specific T790M mutation. Um, and so that mutation can arise and lead to drug resistance down the line. So that leads to ushering in the second generation with afatinib. I can see now this is it's another EGF binder inhibitor, but this now is binding EGFR covalently rather than being reversible like the previous two, which makes it more potent. But I can still see under key limitation here, it's got poor activity. It's still vulnerable to that same mutation. So again, that's informing my own thoughts around what strategy I might want to take with my own molecule. So now I get into the third generation. We're back on osimertinib. This is another covalent binder, but here we're now seeing it's designed to actually bind to that specific mutation site. And because of that, we're seeing it has less affinity for wild-type EGFR, which can help with specificity and avoiding side effects. But now, let's see. Yep. Okay. So it has high potency against this specific mutation that was causing the problem before. But we are seeing that there are remaining limitations. So we can see that acquired resistance still develops due to now there's another mutation. So 7697S mutation that's involved that can arise, and now that's leading to another avenue for drug resistance. So again, really great information to have. And now that is giving me a sense of the challenges that I'm going to need to overcome. And it's another opportunity to go deeper on the information. So continuing to narrow down here on a potential strategy for design of a novel molecule or novel treatment strategy. So now let's see, I'll ask another follow-up. So about this specific C79S mutation to see if this is a strategy that others might be already pursuing, and see that it'll give me a summary of this specific mutation, why it's challenging. I can also see that there are actually a number of novel compounds within the clinic already. There are various different treatment strategies, so single therapy, combination strategies. And so now we're kind of getting into what's the most modern developments, what's the cutting edge, what are those novel therapies that are in trial right now. So now I'd like to get a better sense of what these strategies are for the treatment of NSCLC. So I'm going to actually click over to the detail page, and now we're going to look at detail page for the disease itself. So let me see where if I can go back to my ligand information. I know that NSCLC here is the primary driver. And now I've walked through a ligand detail page, protein detail page. Now here's another page where we're reorienting the context of our information around the disease. So This is a disease page set up for obviously an NSCLC. It's going to give me a drug intelligence summary of all the current treatments that are oriented around the specific disease. It'll give me a lineup of synonyms, again, links to external databases, identifiers, and right on the homepage, we're going to give you a list of all the current therapies that are on the market. And actually, I can sort by phase, so I can even dive into those therapies that might still be in the research phase. They might still be in clinical trials. I can view the entire list right here. So I mentioned that we have a giant collection of disease biomarkers. I can click here and browse through those if I'm interested. But let's go over to the ligands page here. And again, so we've now reoriented our ligand information and showing those that are associated with NSCLC. I can see all those ligands that we know that with known associations. I can click into all the proteins that have known associations with this specific disease. And then I can click the trial information here as well. So I can scroll down here and I can see all the current trials. I can scroll through, get a sense of which ones are, what those strategies might be. I can see a number of antibody strategies, a number of small molecule strategies, a number of combination strategies. So that's all going to be now valuable to me formulating a novel therapy to know what's on the cutting edge, what's in trial, what's in treatment. Okay, so let's see, I can even scroll through here. I can see here's my active trials. I can utilize these filters at my fingertips here to show, okay, now here's the trials that are just active or actually in recruitment now. So again, just trying to get a sense of what's the cutting edge, where are my compatriots, my competitors focusing their efforts, and what strategies are they pursuing. Okay, so I can go back up. I can click the ligands tab here. Again, this is showing me all the ligands that are associated with this disease in particular, get a sense of the therapeutic landscape. I can see there's over 39,000, almost 40,000 small molecule ligand, or actually all types of ligands that we're showing you here. So that's way too many to work with. So I can use some of these filters on the sidebar to try and get more specifically what I'm interested in. So let's say to start with, I'm interested in small molecule ligands. I want them to be fairly well tolerated. So let's choose category 3. I'm interested in those that are both investigational status as well as approved in the US. And then I'm interested in, let's see, let's narrow it down to EGFR as a specific target. And now we've narrowed it down from almost 40,000 ligands to just less than 50. So now this is a really workable list that I can parse through. And again, this just helps me inform my own decision making, how I want to design my own molecule, how I want to design my own treatment strategy. So from there, I can export this list as an SDF file, and then I can take that, share this list of all the current strategies with my team, with any medicinal chemists on the team. I can import this list into other tools that I might have, just continue following down that workflow to get to a specific short list of strategies and molecules that I might be interested in pursuing and hopefully getting a new treatment into the clinic. So what I really wanted to illustrate with this workflow is trying to show how we have these two browsing methods. So we have Newton we have the BioFinder interface, and I wanted to emphasize how really complementary these two methods are. So as I showed you, I'm not spending all my time in Newton. I'm not spending my time only in the interface. I could if I wanted to, if that was the workflow I was following, but really we think that both have their strengths and weaknesses, and they kind of compensate for each other. So, if you've used an AI tool before, some type of large language model based tool, um, you know that this type of response is relatively common. So like if I ask it a question about what are the various immune checkpoint inhibitors, it'll probably just there could be potentially hundreds of different answers if I want to think about the full list, but an AI model like this will probably only pick out a few and display it to you in the response. And so that kind of leaves you with questions where you're thinking, okay, is that the complete list? Is that exhaustive? If not, why did it pick these ones versus the others? So in those cases, a lot of that reasoning that the model is doing is kind of behind the scenes. So in that case, that really makes the BioFinder interface really valuable, so that the interface gives me the freedom to click through, see the full data set, filter down as I'm interested, so I can really see the full picture and then narrow down as are important to me. Um, so yeah, I just kind of wanted to emphasize how flexible we try and make that interface, and present the most flexible tools as possible to you, to get you down and find the answers that you need. So the last thing I want to show here is we've explored a number of small molecule ligands. But let's say I'm actually interested in protein or antibody therapies for NSCLC. So let's go back to Newton here, and I can ask this question as well. Where was I? Okay, so here are there any antibody therapies for NSCLC? And I can see it's going to lay out the primary treatment strategies once again, that antibody therapies are centered around. So I can see there's therapies centered around PD-1 or PD-L1 as a target to affect the immune response. I can see that here's going with the same strategy targeting EGFR. There's a number of antibodies that will follow that strategy. You can see anti-VEGF strategies to try and prevent blood vessel construction, and a number of other strategies as well. So if I can scroll up here, following the theme of targeting EGFR, I can see it lists amivantamab here. That's a bispecific antibody, so that's kind of interesting to me. I can click on the link here. It'll take me to an antibody detail page, and I can view some additional information about this. So I can see here's a cartoon that we've generated that shows the general structure of the antibody, the various regions of the antibody that are important to function. Again, a drug intelligence summary here with approval status, synonyms, and then just general information that would be relevant to this antibody, including the antigens. So I can see it's going to target EGFR as well as hepatocyte growth factor receptor. So let's see what other information we have here. I can click into the sequences tab. It'll give me the full sequences of the specificity domains, so the heavy and light chain sequences. We'll even pull out the CDR sequences on both the heavy chain and light chains, so I can get a sense of what makes this antibody specific and how I can modulate those if I want to adapt it to my own use case. And of course, since it's a bispecific, we have actually two sets of heavy chains and light chains. We lay all of that information out right here. more pharmacology information, so I can look at the various binding assays, and this checks out EGFR, MET, those are the primary targets I'm interested in. ADME information, there's the proteins that we're binding to, associated diseases, and then more clinical trial information. So I can scroll through, see the trials that are involved. So again, just really trying to show you all the information at your fingertips, reorient according to the context that you're viewing. So finally, let's go back to the disease page here. Again, this is showing all the ligands that are associated with my disease of interest. Before I filtered down to narrow down the list of small molecules, now I'm interested in proteins and antibodies. So let's look at that. Let's just look at antibodies actually. So yeah, now I've narrowed it down to a list of just 11. I can export this list. I can explore the other antibodies on this page according to my own interest. So just gives me an idea of the other antibody treatments on the market, how they're constructed, what those treatments or binding strategies are, how they can affect my disease of interest. So just a few examples here of how you can use BioFinder to parse through multiple levels of biology, inform you of your own drug discovery and development workflows. Just wanted to again emphasize that the value that we focused on here is in how we've built out our data domains, pulling them together under the same umbrella, giving you, the users, the ability to explore the data in whatever way makes sense to you, what fits into your interests and into your workflows. So I can start, again, Q&A session with Newton, get up to speed on a given topic, handle more complex queries. I can dive into the interface itself, go back to the BioFinder homepage, start researching on your own, use the search bar here, jump into searching keywords, querying our data directly, navigating to various detailed pages for more specific information. And then again, easily jump back and forth for whatever use case. We give you the right tools to get the job done. So just as a teaser here, you know, we think BioFinder, we've really worked on making it the best it can be, but we're always improving it. So I just wanted to share some things that are coming soon. So Pathway Explorer, I briefly showed some pathway information that's already in the platform, but we're working on really building this out, making it a lot more full-featured, allowing you to search and view pathway summaries, giving you the full diagrams of the pathways these proteins are, your protein of interest might be involved in. Second, we're bringing a whole new data domain around RNA therapeutics to the platform. We're going to view the details, allow you to view the details, structures of these therapeutic molecules. Third, we're bringing Newton right now, as I showed you, kind of Newton and the BioFinder interface are kind of in separate areas of the application. But what we're going to do now is integrate those more and more so I can be viewing some information within the BioFinder interface, but I can have Newton present on the sidebar that knows the page that I'm looking at, and I can chat with Newton about the specific context that I'm viewing right away. Fourth on the list, binding pocket similarity. So we're working on some really advanced analytics that will allow me to compare active site binding, structure and composition between different proteins. And finally, we're working on adding even more sophisticated safety information to allow you to delve more deeply into safety, toxicity, reference information, as well as predictions.